Welcome back to another Sleepy Steve story. Before we drift off tonight, remember to subscribe to help Steve-O out, and I will keep making more sleepy stories to help you study gently, revise quietly, and settle your mind at the end of the day. Tonight, we are moving gently into Unit 3, Area of Study 2, where biology starts to feel a little brighter, a little more energetic, and yet still calm enough to carry us towards sleep. In this part of the course, we are thinking about how cells gain and transform energy. We are thinking about life not as a still thing, but as a constant flow of matter and energy, moving from the environment into cells, moving through chemical pathways, and being stored, released, and reused in ways that keep living organisms alive. For this episode, we are going to settle into photosynthesis. We will not rush it. We will let the idea unfold slowly. Photosynthesis is the process by which plants, algae, and some bacteria use light energy to convert carbon dioxide and water into organic molecules such as glucose, while releasing oxygen as a byproduct. That sentence contains a lot, so let us soften it and walk through it piece by piece. Photosynthesis matters because it is one of the main ways that energy enters most ecosystems. Light energy from the sun is captured by photosynthetic organisms and transformed into chemical energy stored in the bonds of glucose and other carbohydrates. Those carbohydrates can then be used by the plant itself, or passed through food chains when organisms eat plants or eat organisms that have eaten plants. So when you study photosynthesis, you are not only studying a plant process, you are studying a biological doorway through which energy flows into living systems. A useful exam sentence might be this: photosynthesis transforms light energy into chemical energy by using carbon dioxide and water to produce glucose and oxygen. That is simple, clear, and accurate. Then, if you need to extend it, you can say that this occurs in chloroplasts and involves two linked stages: the light-dependent stage and the light-independent stage. Let us begin with the site of photosynthesis. In plants, photosynthesis occurs in chloroplasts. Chloroplasts are organelles found in the cells of plant tissues that carry out photosynthesis, especially in leaf cells. They have a double membrane, an internal fluid-filled space called the stroma, and stacks of membrane-bound structures called grana. Each granum is made of flattened sacs called thylakoids. The thylakoid membranes contain chlorophyll, the green pigment that absorbs light energy. You might like to picture the chloroplast as a quiet green workshop inside the cell. The thylakoid membranes are like folded workbenches where light is captured, and the stroma is like the open workspace where carbon compounds are built. This image is simple, but useful. The light-dependent stage occurs on the thylakoid membranes. The light-independent stage occurs in the stroma. Chlorophyll is central because it absorbs light energy. Chlorophyll does not absorb all wavelengths of light equally. It absorbs strongly in the blue and red regions of the visible spectrum and reflects more green light, which is why many leaves appear green. When chlorophyll absorbs light, electrons within chlorophyll molecules become energized. This is the beginning of the energy transformation that drives the first stage of photosynthesis. Now let us move into the light-dependent stage. The light-dependent stage requires light. It occurs in the thylakoid membranes. Its main job is to capture light energy and use it to produce energy-carrying molecules, mainly ATP and NADPH. It also splits water, releasing oxygen. ATP stands for adenosine triphosphate. It is often described as an energy carrier in cells. NADPH is another energy carrier, holding high-energy electrons and hydrogen that will be used in the next stage. For VCE biology, it is helpful to remember that the light-dependent reactions produce ATP and NADPH, and that these are then used in the light-independent reactions. In the light-dependent stage, light energy is absorbed by chlorophyll in photosystems. This energy excites electrons. The excited electrons move through an electron transport chain in the thylakoid membrane. As the electrons move, their energy is used to help pump hydrogen ions across the membrane, creating a concentration gradient. Hydrogen ions then flow back through ATP synthase, an enzyme that helps produce ATP from ADP and inorganic phosphate. This process is called photophosphorylation because light energy is being used to make ATP. That may sound technical, so let us say it gently again. Light excites electrons. Excited electrons move through a chain. Their movement helps create a hydrogen ion gradient. The flow of hydrogen ions through ATP synthase produces ATP. The energy of sunlight has now been transformed into chemical energy in ATP. Water also has a key role. During the light-dependent stage, water molecules are split in a process called photolysis. The splitting of water provides electrons to replace those lost by chlorophyll, provides hydrogen ions, and releases oxygen as a waste product. This oxygen diffuses out of the leaf and into the atmosphere. So the oxygen that many organisms use for aerobic respiration is connected to the splitting of water in photosynthesis. The light-dependent stage therefore has clear inputs and outputs. The inputs include light energy, water, ADP, and inorganic phosphate, and NADP+. The outputs include oxygen, ATP, and NADPH. Oxygen is released. ATP and NADPH move into the next stage. Now we can drift into the light-independent stage. The light-independent stage does not directly require light, but it depends on the products of the light-dependent stage. It occurs in the stroma of the chloroplast. This stage uses carbon dioxide, ATP, and NADPH to build carbohydrate molecules. It is often called the Calvin cycle. The name light-independent can sometimes be misleading. It does not mean the stage can keep going forever in darkness. It means that light is not directly absorbed in this stage. However, because the light-independent stage relies on ATP and NADPH from the light-dependent stage, it is still indirectly dependent on light. Without light, ATP and NADPH production slows or stops, and so the light-independent stage cannot continue for long. In the Calvin cycle, carbon dioxide is fixed. Carbon fixation means that carbon dioxide from the air is incorporated into an organic molecule. The enzyme Rubisco plays a major role in this process by helping carbon dioxide combine with a 5-carbon compound through a series of enzyme-controlled reactions. ATP and NADPH provide the energy and reducing power needed to form carbohydrate molecules. Some of the carbon compounds produced can be used to make glucose, starch, cellulose, and other organic molecules. For exam purposes, you do not need to recite every step of the Calvin cycle in the same detail as a university biochemistry course. What matters is that you can explain the overall role of the stage. The light-independent stage uses carbon dioxide, ATP, and NADPH to produce carbohydrate. The ATP provides energy. The NADPH provides high-energy electrons and hydrogen. Carbon dioxide provides the carbon atoms that become part of organic molecules. Let us bring the two stages together. The light-dependent stage captures light energy and produces ATP and NADPH. The light-independent stage uses ATP and NADPH to fix carbon dioxide and produce carbohydrate. The stages are linked because the products of the first stage are required by the second stage, and the second stage returns ADP, inorganic phosphate, and NADP+, so that they can be reused in the light-dependent stage. This cycling is a beautiful idea in biology. Molecules are not simply used once and forgotten. They move through pathways. They are converted, recycled, and reused. ATP becomes ADP and phosphate when energy is released. NADPH becomes NADP+ after donating high-energy electrons and hydrogen. These molecules then return to the light-dependent stage, ready to be energized again. The overall word equation for photosynthesis is carbon dioxide plus water, in the presence of light and chlorophyll, produces glucose plus oxygen. The balanced chemical equation is 6 carbon dioxide molecules plus 6 water molecules producing 1 glucose molecule and 6 oxygen molecules. In symbols, that is 6CO2 plus 6H2O gives C6H12O6 plus 6O2. It is useful to know the equation, but it is more important to understand what it represents. Carbon atoms from carbon dioxide become part of glucose. Oxygen gas is released mainly from water. Light energy is transformed into chemical energy stored in organic molecules. Now let us think about why photosynthesis is described as an anabolic process. Anabolic reactions build larger, more complex molecules from smaller molecules, usually requiring an input of energy. Photosynthesis builds glucose and other organic molecules from carbon dioxide and water using light energy. So photosynthesis is anabolic. It stores energy in chemical bonds. This contrasts with cellular respiration, which we will explore in the next episode. Cellular respiration breaks down organic molecules to release energy in the form of ATP. Photosynthesis stores energy. Respiration releases usable energy. In plants, both processes occur. Plants photosynthesize in chloroplasts and respire in mitochondria. They are not opposites in a simplistic way, but they are linked within the larger movement of matter and energy through living systems. Now we can consider factors that affect the rate of photosynthesis. In biology, a rate refers to how quickly a process occurs. The rate of photosynthesis can be influenced by light intensity, carbon dioxide concentration, temperature, and water availability. Light intensity matters because light provides the energy for the light-dependent stage. When light intensity is low, increasing light intensity can increase the rate of photosynthesis because more light energy is available to excite electrons. But after a certain point, another factor becomes limiting. The chloroplast cannot keep increasing photosynthesis forever just because there is more light. Once the photosystems and enzymes are working near capacity, or once carbon dioxide becomes limiting, further increases in light may have little effect. Carbon dioxide concentration also matters because carbon dioxide is required in the light-independent stage. If carbon dioxide levels are low, the Calvin cycle cannot fix carbon quickly, and carbohydrate production is limited. Increasing carbon dioxide concentration can increase the rate of photosynthesis up to a point, but again, another factor may eventually become limiting. Temperature affects photosynthesis because many steps in photosynthesis are controlled by enzymes. At low temperatures, enzyme activity is slower, so the rate of photosynthesis may be lower. As temperature increases, enzyme activity usually increases up to an optimum temperature. Beyond the optimum, enzymes may begin to denature, changing the shape of the active site and reducing their function. This can decrease the rate of photosynthesis. Temperature effects are especially important for the enzyme-controlled reactions of the Calvin cycle. Water availability matters because water is an input in the light-dependent stage and because water loss affects stomata. Stomata are pores in the leaf that allow gas exchange. Carbon dioxide enters through stomata and oxygen and water vapor can leave. When water availability is low, plants may close their stomata to reduce water loss. But closing stomata also reduces carbon dioxide entry, which can limit photosynthesis. This brings us to the idea of limiting factors. A limiting factor is the factor that is in shortest supply relative to need, and therefore restricts the rate of a process. In photosynthesis, light intensity, carbon dioxide concentration, temperature, and water availability can all become limiting factors. In an exam, if you are interpreting a graph of photosynthesis rate, look for the factor that is preventing the rate from increasing further. For example, if increasing light intensity increases photosynthesis at first, then the graph plateaus. Light is no longer the limiting factor. Something else, such as carbon dioxide concentration or temperature, may now be limiting. If carbon dioxide concentration is increased and the rate rises again, then carbon dioxide was limiting. This style of reasoning is important. Biology is not just about naming factors, it is about explaining relationships. Let us now connect photosynthesis to plant structure. Leaves are adapted for photosynthesis. They often have a broad, flat surface area to capture light. They contain chloroplast-rich palisade mesophyll cells near the upper surface. They have spongy mesophyll with air spaces that allow gases to diffuse. They have stomata and guard cells to regulate gas exchange. They have xylem to transport water to the leaf and phloem to transport sugars away from the leaf. Structure supports function. When students write about photosynthesis, one common mistake is to say that plants photosynthesize to make energy. A more accurate statement is that plants use light energy to produce glucose, storing energy in chemical bonds. Energy is not created, it is transformed. The plant can then use glucose as a substrate for cellular respiration, or convert it into starch for storage, cellulose for cell walls, or other organic molecules required for growth. Another common mistake is to say that the light-independent stage does not need light at all. A more careful answer is that the light-independent stage does not directly use light, but relies on ATP and NADPH produced by the light-dependent stage. That wording will help you avoid oversimplification. A third mistake is to confuse the source of oxygen gas. The oxygen released during photosynthesis comes from the splitting of water during the light-dependent stage, not directly from carbon dioxide. Carbon dioxide provides carbon atoms for carbohydrate production. Water provides electrons and hydrogen ions, and its splitting releases oxygen. So, if you wanted to build a strong VCE explanation, you might say, photosynthesis occurs in chloroplasts and involves a light-dependent stage in the thylakoid membranes and a light-independent stage in the stroma. In the light-dependent stage, chlorophyll absorbs light energy, water is split, oxygen is released, and ATP and NADPH are produced. In the light-independent stage, carbon dioxide is fixed using ATP and NADPH to form carbohydrate. This transforms light energy into chemical energy stored in organic molecules. Let us now slow the idea right down. A leaf rests in the light. Inside its cells, chloroplasts hold folded thylakoid membranes. Chlorophyll absorbs sunlight. Electrons are excited. Water is split. Oxygen drifts away. ATP and NADPH are formed. Then, in the stroma, carbon dioxide is drawn into a cycle of reactions. Carbon is fixed. Carbohydrate is built. Energy that began as light is now held quietly in chemical bonds. That is the heart of the episode. Photosynthesis is not simply a formula. It is an energy transformation. It is a series of linked enzyme-controlled reactions. It depends on structures inside chloroplasts, on light and water and carbon dioxide, and on environmental factors that can increase or limit its rate. Before we finish, let us review the key links one more time. Chloroplasts are the site of photosynthesis. Thylakoid membranes are the site of the light-dependent stage. The stroma is the site of the light-independent stage. Chlorophyll absorbs light energy. Water is split, releasing oxygen. ATP and NADPH carry energy and reducing power to the Calvin cycle. Carbon dioxide is fixed into organic molecules. Glucose and other carbohydrates store chemical energy. And finally, remember the exam verbs. If you are asked to describe photosynthesis, give the process and the main inputs and outputs. If you are asked to explain photosynthesis, link the stages and show how one stage supports the other. If you are asked to analyze a factor affecting photosynthesis, identify the factor, explain how it changes the rate, and discuss limiting factors. If you are asked to compare photosynthesis and respiration, focus on energy transformation, sites, inputs, and outputs. So now, as the evening settles, let the details arrange themselves. Light-dependent stage, thylakoid membrane, chlorophyll, water, oxygen, ATP, NADPH. Light-independent stage, stroma, carbon dioxide, Calvin cycle, carbohydrate. Energy transformed, matter rearranged, life quietly maintained. That is enough for tonight. You have followed the light into the chloroplast, watched it become chemical energy, and traced the beginning of the flow of energy through living systems. Let those ideas rest now. Let the leaf grow still. Let the cell become quiet. And let your mind drift gently towards sleep.